Hello, everyone. My name is Claudia Mello, and my colleague Nayet Conejal and I will be presenting the antiviral activity of Psidium guajava against herpes simplex type 1. Guava is a plant that is cultivated in warm tropical climates all over the world. And leaves, roots, and fruit has been traditionally used to treat illnesses. The objective of our research project is to study the in vitro antiviral activity of commercial samples of guava leaves against herpes simplex type 1.
The herpes simplex type 1, HSV1, belongs to the family Herpes viridae. Its subfamily is alpha herpes viridae. It is a double-stranded DNA virus with a diameter of about 225 nanometers. In our material and methods, the first thing that we did was prepare our extract with a commercial sample from the brand Cupa. We measured about 1 gram in a 10 ml of 60% ethanol and evaporated, then resuspended it in 20% DMSO, dimethyl sulfoxide. Afterwards, we tested the cytotoxicity of the extract using an XCT assay. We needed to know if the extract was in fact toxic or not to the cell. In addition, we use a plaque reduction assay to test the antiviral activity against herpes simplex type 1. In this experiment, HSV is treated with different concentrations of guava leaf extract and compared to a virus control.
Okay. So what are you seeing in the first graph? The red color line is that the guava extract is not toxic to the cell. The two highest extract concentrations show some toxicity, but as soon as we dilute further, we see 100% of cell viability. With this dose response curve, we can get a value known as CC50 or half maximum concentration that kills 50% of the cells. Our CC50 is around 15 milligrams per ml. That means you need a high concentration of the extract to kill 50% of the cells. The green color corresponds to the antiviral activity. We were not able to produce an optimal dose response curve to calculate the half maximum effective concentration, EC50, that blocks 50% of the virus. But even the lowest concentration tested protected about 80% of the cells. About 20% of viral replication can be seen. All we can say is EC50 is lower than 20 micrograms per ml. Now that we obtained these values, we can calculate the ratio between the CC50 and EC50 values. This is the therapeutic index or TI value. If we get a value above 10, it means that there's a true antiviral activity. In our results, we obtain a TI value higher than 750. This is impressive because values above 100 indicate that future studies should be done because the molecule tested has a strong antiviral activity. The control molecule for the antiviral activity was carrageenan, and the dose effect curve showed an EC50 of 85.7 nanograms per ml, with a 95% confidence interval between 78 and 92.9. That was the expected value for our control. Also, we had a control for the cytotoxicity assay. We used twin 20, and we obtained the expected result. These values just tell us that the experiment was accurately performed. So in conclusion, Cupa extract had an EC50 value lower than 20 micrograms per ml against HSV1. The therapeutic index was higher than 750. Future experiments should explore the leaf's chemical composition and the potential mode of action against HSV1.